In search of soil.
If you look at I'll call it the old school approach or a lower tech approach, if let's say you had any biological product, anything out there, in the past, it would just be you'd have to either apply it as a seed coat and then plant the seed with it already on it, or you'd have to plant it and then spray the soil surface and let it percolate through and hope it makes contact. Looking at both those options, a seed coat and say a soil surface spray, when it comes to biologicals, what are the limitations of those application methods?
Sure. And when I talk about biologicals, recognize there's a huge umbrella of biological products. And what I'm talking about really is the microbial biologicals. So these are living, they include either bacteria or fungi. Um, and some of the challenges there with, say, for a seed treatment is, for treatments that are done way by manufacturers way up in the supply chain, right? That's months in advance that the microbes are applied to the seed, and that can be challenging to maintain viability of those microbes on the seed for many months potentially. So that's one challenge, and it's a limitation because it limits the types of microbes that are being put on as seed treatments. Primarily, it limits it to in the case of bacteria that form spores. So these are bacillus strains, which are most commonly found as seed treatments. But there's a whole wide range of different bacteria that don't form spores, and it really limits their ability to be applied as a seed treatment back at the manufacturer, right? In terms of the farmer, with seed treatments, you have a lot of chemistry on there as well. So, whatever the seed package is, you're kind of stuck with what that is. So the ability to apply a biological later, potentially closer to time of use, can widen the range of microbes that are possible to put on the seed. You do have to be mindful of the chemistry that's on the seed to make sure that there's compatibility, that it's not going to harm the microbes. But it gives the farmer a lot more flexibility if they're applied closer to time of use versus being treated far in advance. The other, like I had talked about in furrow delivery, it gives the farmer a lot of flexibility. So they're not having to make decisions on seed treating well in advance. They can make decisions, as they go out that day, right? What are they going to put in their tank mix, and gives them flexibility on biologicals as well as, insecticides, fungicides. So there's kind of, you have to understand both the impact on the microbes, but then also the flexibility to the farmer for using these types of products.
When you look at plant growth enhancing microbes or beneficial microbes that are either maybe they help reduce disease pressure, maybe they help actually stimulate growth, how much does that spectrum open up when you can go to a just-in-time application approach versus a spore seed coat approach?
It opens up tremendously, and this is where you look at decades of research in finding these plant growth promoting bacteria and fungi, but very limited commercialization. And part of that reason comes back to the stability of the living microbe, and by being able to use them closer to the farm gate where they're haven't been put on as a seed coat, that can open up the number of bacteria. And a lot of the discovery work that happens, and this is at universities, at companies big and small, there's a tremendous amount of discovery work going on right now, and roughly 3/4 of the bacteria, just kind of through discovery work, are what's called gram-negative bacteria. So if you go into the research literature and you look, nearly 3/4 are these non-spore forming bacteria, and the reason is they're so ubiquitous in the soil. They're everywhere. They associate with the plants readily. So when you're doing, you're looking at the soil and the plant and the bacteria that's associated with it, what you find is a lot of these bacteria that are hard to get into a product form. And so, that's one of the reasons the company that I work with, 3-Bar Biologics, we wanted to find a way to better deliver these microbes so that more of them that were discovered in research could be made into product, because they're just so hard to get into a stable product form. But we're seeing more and more of these microbes being discovered. And so how do you work on getting them into a product form? And that's really kind of the Achilles heel of the industry right now, in an area we're working in.
In 3-Bar, the company you work for, 3-Bar Biologics, that came out of OSU and a lot of the research that was being done there. Can you talk about some of the research that was taking place at Ohio State and how that led to the company?
Yeah, sure. And so, like I said, this discovery research, it usually starts with, soil samples from plants, and you start to look at, what's associated with it. And then you screen out microbes that can really provide a good benefit to the plant, right? And through this work at Ohio State, and there's, many, many land grant universities in the United States and all over the world that are doing this kind of research. And several researchers at Ohio State had found, a number of strains that could provide benefit to plant health. They were all gram-negative, non-spore forming bacteria. Like I had said, these bacteria that are really hard to get into to stabilize. And so it was through that kind of realization of, all this great research is being done, but, products aren't being developed. We're not getting these great this great potential from the microbes into the hands of farmers. And so that was really kind of why the company was founded, was on the premise of, there's got to be a better way to get these microbes into a product, into the hands of farmers. And so we developed a technology to do that. We call it our live microbe technology. But it was really out of that realization that, here's these great microbes, but to get them to the farmer, we're going to have to do this a different way.
How challenging is it to just multiply out those microbes? So they exist in the soil, they're beneficial. And even if we forgot commercialization and we just said, hey, I have a lab here, or I have the most basic equipment that any farmer would have possible. How easy is it to take those and multiply out the ones you want and then just get them into a large enough quantity that you can then go apply them on the field? Even if we take out, like, hey, we're not trying to sell it, we're not trying to commercialize it. What I'm getting at is, I think a lot of farmers would love to do that. How complex actually is that?
Yeah, so it sounds easy. You can grow a lot of microbes, but maybe not the ones you necessarily want. So, one of the challenges in, growing microbes is making sure that you're doing it in a what we call aseptic manner. So you're doing it in a way that you're starting with everything sterile, you're adding the microbes that you want and growing them. And in the lab, that's, fairly straightforward to do. Those trained in microbiology do it quite well. But for a novice or somebody that's not familiar with the art, it can be more challenging because you can contaminate something so easily. And so in general, it can be done pretty straightforward in a lab environment, but you get out on the farm, you're in an open system, it becomes more challenging to maintain, to maintain a pure culture, if you will.
So you don't really know what you're getting. You're probably multiplying something, what that is, right? Like,
right. It could be pathogens, right? So you don't want to multiply pathogens. So yeah.
One microbial product that I think a lot of farmers have had access to in the past, it's like a powder, whether it's myco something usually is in the name, and there's a powder there. Are those the non-gram negative bacteria? Are those the spore forming bacteria? That's what would be in those powders?
In some cases, yes. And thank you for bringing up the dry formulation, because the microbes will survive. They basically get put into a dormant state. So when they're dry like that, you kind of put them to sleep. And so both the spore form as well as the non-spore forming bacteria can be made into a dry formulation and be fine at room temperature for a period of time. So that's actually a really good strategy for formulation and developing a product. But it can be very expensive. Anytime you're doing further processing beyond just growing the microbes, from a manufacturing standpoint, it can get more and more expensive. And so as a finished product, those products, if they're not using kind of lower tech technology to create the dry formulation, they can be kind of priced out of the market, if you will.
What about efficacy in the field of a liquid applied product versus a dry formulated product? Do you think there's a difference there?
Well, back to, the microbes are in a dormant state when they're formulated dry. Some of those products are wettable powders, so they'll be brought back into a liquid and then applied. So there can be some kind of coming out of that dormancy, some lag there. But in general, they should work similarly, particularly if they're both delivered as a liquid, if it's a wettable powder and brought back into a liquid and delivered as a liquid. But one of the things to keep in mind, the extra handling involved with with a dry formulation sometimes isn't ideal. Farmers usually like having a liquid; they can just add it to their tank mix. It's a little bit more readily usable. But in general, they both work just fine.
I know from just looking at what 3Bar's been doing, a lot of these microbes originated, you said, right out of an Ohio field, and they were isolated out of there. One concern that I know people have, and this came up in episodes, and other people, scientists have various thoughts on this about local efficacy of microbes. So you get a microbe out of Ohio, and I want to use it here in California, or I want to use it in Florida, or I want to use it in Maine. Is it going to work the same in all conditions, or are they that specific where an Ohio microbe out of that field is going to work best in that field in Ohio?
There is some regionality, and it really comes back to, I think, understanding that not every microbe is meant to be a silver bullet where it works everywhere all the time. Some that are coming out of conditions that, say, are cooler, wetter, will probably do better going into conditions that are cooler and wetter, and vice versa. Strains that are isolated out of places that are more arid, maybe have less drier conditions, probably do better in those types of conditions. So there is some dependency there, but microbes, they can work across. So I think trying at least at a regional level to make sure that they work across some different climates is important. And also the function that they give. You really, every microbe it's not going to solve all of the problems in the field, but in targeted ways they can help. Say, for instance, help with nutrient availability or help the plant through certain abiotic stress. You have to kind of understand where you're applying it and why, and know that it's not going to be something that's going to solve, all of the problems in your field, but it could help under certain conditions to improve your yield in certain areas under certain conditions.
That's a good point that you brought up there, because I think some people expect, a dramatic transformation, like this weak, staggering plant. You apply it, and boom, you have a redwood growing all of a sudden there. When it comes to realistic expectations, what should someone think about if they're considering applying a beneficial growth microbe?
Yeah, so certainly under more controlled environments, you can set up experiments where you can see kind of a half dead plant and one that had microbes applied to it, is growing quite well. But out in a field under, conditions, varying conditions, you may not see that same kind of direct response. But for one of the things that we see with a microbe we've been using that was discovered at Ohio State, it works really well under conditions where, if you want to go out and plant earlier, it's helping that plant through kind of that early abiotic stress when it's cooler and wetter. So in that environment, this microbe really works well, and you can see some of that immediate early season kind of vigor when the bacteria are used versus when they're not. So there's a case where certain conditions, this microbe can help the plant through that kind of stress, and you can see it in the early vigor. Will that translate to a huge increase in yield at the end of the season? Depends on the season, but you can at least say I saw an early vigor response. It helped the plant through some of that early stress. And so, you have to think about, understand how these products work, that they can help under certain conditions, but not expect that they're going to give you, a 50% bump up in yield. It doesn't quite work out that way, but using them can help in general. It's kind of, we like to think of it kind of as a insurance policy, that if the conditions are more detrimental, whether that's some early season flooding or some conditions that were harder to get the stand up, they can help the plant through those conditions. So it's kind of like a de-risking, if you will.
Sure. But if you look at all the factors that affect a crop, a really dry year, a really wet year, pathogens, yield increase, is there one area that you say, okay, like this is really a sweet spot for microbes? Like if you look at all the research, microbes can definitely really help here. And then once you start getting out into other areas of yield increases, drought tolerance, whatever it is, they have less of an effect. Like where's the sweet spot for microbes at this point, do you think?
Yeah, so the ones that have been isolated, we call them kind of the wild types, the naturally derived, right? So they came out of the soil or from plant material, and those microbes, they do, you kind of have for plant, as a plant growth promoting microbe, kind of the laundry list of things that they can do, are fairly consistent, if they're plant beneficial microbes. So they help with, you think of these microbes, they're little chemistry factories, if you will. So, as they associate with the plant and are fed by the plant, that's the relationship that forms. They're basically producing a wide range of different chemistry. So these metabolites that they're producing, and these could be plant hormones. They can be, different organic acids. And so it triggers this response in the plant as well. And so that's where that association happens, that can help the plant through the abiotic stress, whether that's, not enough water, too much water, through some flooding event, too hot, too cold, and so forth. And also, and they can be antagonistic to other pathogens, right? So they can help with some of the biotic stress as well. But all of this kind of general help these microbes provide. But it does depend a little bit on the strain, a lot on the strain in terms of what it will, what genes it has. So its genetic makeup, and what maybe it produces more organic acids. So it really can help in this case with maybe solubilizing phosphorus, or it produces certain enzymes that help with, say, nitrogen fixation. So what research, what's happening in research is trying to screen out and find kind of your super performers that have those genes that are producing, certain enzymes or certain, plant hormones or so forth, to have better microbes. So that's what we're seeing kind of in the pipeline, are better microbes. But these are all naturally derived. So, when you hear, I know, one of the big complaints about using living microbe products is the variability. You put them, into the field, and depending on, there's such a wide variety of variability in the field, that the microbes, they have certain triggers where they're producing certain chemistry. And so that variability kind of compounds on itself. So in some cases, they may not be triggered to produce a certain chemistry. So those are the challenges, and kind of how those challenges are being overcome is through really through some of the biotechnology that is, we've seen some of it on the plant systems, right? We're now seeing that with microbial systems where you can do genetic engineering to make sure that the function that the microbe's supposed to be providing, it's going to provide that no matter what conditions it experiences when it's put in the field. So that's kind of the next generation of products. We're starting to see some of them. Certainly they're being researched, but that's where we're going to have better and better microbes because of that.
Do you think, if we go to the question, somebody might say, well, why do we even need these? Like, if they're already there, shouldn't they already be proliferating in the system? How much do you think agricultural methods and management practices affect these field microbes? For instance, if you go back in the research, is there a difference between, say, no-till and till? Is there a difference between continue coverage where you have a cash crop and a cover crop versus where you're having fields just sit fallow, where there are more favorable conditions to have these microbes just proliferate in the soil? So sure, you can add them is more of an insurance, but you're not starting from such a depleted state given some methods. Is there a tie-in between management practice and what they're seeing in the field?
Yeah, absolutely. But you have to understand kind of the context of adding them into the field as a precision application, which we kind of talked about there at the beginning, versus really kind of improving your soil health, right? And that's what's getting a lot of attention, and for good reason. In soil health, the idea is that we're getting the biology back into the field. So increasing the soil organic matter, making sure that you have a very diverse and active biology, microorganisms as well as macroorganisms like worms, things like that. So you have very active biology. So that's the goal. And in a smaller system, it's very achievable. In more conventional, larger, broad acre agriculture, it's achievable. It just takes a lot more. There is a good bit of management behind it. So the practices, you touched on tillage, right? In conventional practice, we've disrupted the soil a good bit. So tillage is one thing. Adding, fertilizers, synthetic fertilizers, and pesticides. It all disrupts that soil environment. But you want to be able to, get a certain amount of biology back and continue to work on improving your soil organic matter. And in broad acres, like I said, there's strategies to do that. Certainly practices that are getting more attention, which is great. The whole area of regenerative agriculture, which is kind of the buzzword around this right now. And so some of those strategies involve, either no-till or, strip till, much reduced tillage than what has conventionally been practiced. Also cover crops. So, the goal is, if you're going to have the biology getting put back, you're going to have an active root system there all the time, which is where cover crops can really provide benefit. And then, adding different microbiology sources. So, manures, a great source of very diverse microbiota. So, how do you get more of that into the soil, I think, is the challenge, especially on a broad acre. Mm-hmm. With microbial products, or any biological product, but microbial products, the idea, you could try to more broadcast them, but you have such a limited amount that you're trying to get into the soil that the precision application is where these products are more fit better in terms of existing practices, so that you're just putting it with the plant so they can provide that direct benefit to the plant.
If you went with a microbial product, and let's say you had no constraints on application, like you could full access to the crop from seeding to harvest, and we're throwing economics out. Do you think that if you looked at the whole suite of microbial products, that, let's say it was corn, that there could be X amount of applications of microbials throughout the corn's life to get a significantly better yielding crop if you didn't have those constraints?
Yeah, no, that's a great question. Certainly, depending on, the function that the microbe's trying to provide. So, with corn, nitrogen's a big focus, and there are nitrogen fixing microbes that can certainly help that. And can they provide the nitrogen throughout the growth cycle of the plant at an amount that you're not having to bring in, nitrogen through, foliar application or whatever, which is traditionally done through the season. You could apply other microbes to provide other functions at certain time. I don't think I can fully answer that question. I think it raises a good point, but there's certainly the ability to reduce some of the synthetic chemistries we're applying with the biological products and do it in a way where you're providing at different time points benefit to the plant to help kind of, the goal with this is step change, right? That we're able to have a step change in yield, reduce inputs to be more sustainable. And, biological products, have gotten a lot of interest and excitement over the last few years because they're seen to be able to potentially provide that step change.
Mm-hmm.
But how they're delivered, when they're delivered, the research, it's just, there's not a lot of research to get to the reduction to practice. And a lot of times that's, the discovery work's important, right? We need to discover and the interactions with the plant and the discovery that's going on there. That's critical. But then also reducing it to practice in a way that you can start to see some of these changes is, I think equally as important to make these products work better.
When it comes to precision application, if you look at something like foliar feeding, how effective is that at delivering a microbial product to a plant? When a plant absorbs microbes through its leaves, or it gets microbes on the surface of its leaves, I guess, how effective is that in doing that? Is it, if you apply 100 are taking? Is it you apply 110 take? Where is it on the scale of being effective?
Yeah. So it depends on what you're trying to achieve. There are a couple foliar applied products that I'm aware of that are very effective or can be effective. Some produce hormones, plant hormones. And so they associate with the plant. They kind of, they can grow into the plant. They're called endophytes. So they associate with the plant and get into the plant tissue. And then they can produce things like auxin, which can really help with stimulate plant growth. And there's some other microbes that do different things, but that's one that's common. And that can be very effective at certain growth stages if you're trying to accomplish, stimulate that growth. Biopesticides, we haven't really talked about that, but biopesticides, depending on where the pest is, they may need to be delivered onto the foliar tissue or potentially into a bud or somewhere where that pest might be. And so they can be very effective. The challenge with anything kind of, foliar or above ground is you then have, UV, the sunlight. So you do have to be mindful of how you're delivering that the formulation to make sure that the microbes are protected a little bit so that they're not being killed by UV. But some of that has already, been worked on through additives you can include in your tank mix and so forth in the formulation. But there are products out there certainly that work, that are effective, that aren't just applied with the seed.
I don't know if you've came across this in the precision ag space, but is there enough knowledge out there now on foliar feeding? So let's say we're moving away from biologicals for a quick sec, and I just want to apply nutrients to a plant. Uptake of nutrients through leaves versus uptake of nutrients through roots. Is there a difference there? And when you do precision ag, if you have to apply to foliage, are you going to get the same benefit as if you were injecting it to the roots or drenching the soil? Do you know that?
That's a little bit outside of my expertise. I know, again, back to corn, some, farmers will go out and apply a foliar nitrogen to, get it that boost of nitrogen at a certain growth phase. So I don't know the efficiency necessarily, but when you think about delivery, spraying, anytime you spray a liquid, it's one of the most inefficient processes. You lose a lot through, just off target movement. So from an efficiency standpoint, the more precise your delivery to versus a, more spraying can get better, further coverage potentially, but you can lose a lot too. So you got to think about, how you're delivering it, feeding it. It's challenging in broad acres because you have a lot of ground to cover, right? So spraying's pretty typical with liquid material. When you're going in furrow, you have a little bit more control, right? You can control it right into that furrow, but a good bit's wasted in furrow too, unless you're, there are some new technologies that are turning on and off between the seeds. So you don't waste, if you're just laying down liquid between the seed, you're wasting a good bit of material. So there are new equipment coming out to help kind of be less wasteful. But
Well, with a lot of biologicals, is the waste truly waste? If you put it in an area in between seeds, I think, okay, there's microbes in the soil. They'll just either go dormant in the soil or they'll just wait for the roots to eventually make their way over there. How much of that is actually wasted when you think about a microbial product? Do you need it right up on the root?
yeah, so great question. The microbiology that's in the soil, if you have good biology happening in your soil, you probably have a decent amount of root. The food for the microbes is there. With with root systems, it's the carbon and the exudates from the plant roots that are feeding the microbes. There's other material in the soil too, other carbon in the soil that can help feed the microbes, but they're gonna, there's a population dynamics. So when there are roots around, they increase, and then they eventually tail off once those maybe those roots aren't there. So to maintain it, when we apply a biological product, maintaining, most those microbes will maintain with the root system, but then, when you harvest the plant, they kind of go away. So you're not changing the soil microbiome with what you added. If you added them to, say, the entire field, right, not just with the plant, you're adding such a small amount, and then they're competing with what's there, that they're probably not making a big impact either. And so that's where, you really have to, to improve your soil organic matter, improve the diversity of the microbes. It's really, it takes time. It doesn't happen overnight, right? It takes repeated applications, whether that's manure, putting cover crops in, which cover crops, those seeds will introduce some microbes, and then, you're establishing a population, you're helping that what is there continue to maintain and increase. So it does take a good bit of management and effort over time to make that community come back, right?
Is part of the idea behind some microbial products not reproducing in the soil that they need a specific point in a plant's life to actually exist, and once that point passes, like that's their window, it's over? As I think this is where a lot of people get hung up. They think, well, I
Yep.
put this in, I have a live plant growing there, let's say it's a cash crop, I follow that up with a cover crop, I still have a living root in the soil. Why do those microbes suddenly die off? Why don't they proliferate, and why aren't they multiplying? If they're originally soil born, they're not modified to just, die after 90 days. Why don't they stay in place, and why can't they naturalize and breed out?
Yeah, no, it's a great question. And kind of the way I think about it, and I kind of segment it, is, you have, what's there in a broad, in the bulk soil, right? And there is microbiology there, even, conventional fields that have been, used chemical fertilizers for years. That diversity and kind of level of microbes in that more conventional field that's, use tillage and synthetic fertilizers is going to be reduced. You're not going to have the same diversity. You're not going to have the same level of microbes than one that is, really working on improving the soil organic matter, improving the soil health. And what happens when you add, whether it's multiple microorganisms, there's products that are, we call them consortia, but they contain, multiple different species. You could apply a product like that. You could apply a single species. When you're introducing that, the level that you would need to introduce in a bulk soil to really make a difference would be very high. And that would be something that's pretty expensive to do. And like I was saying, ways that you could introduce more roots in the ground with things like cover crops, that can help improve, especially if you have a diversity of those cover plant species. That helps too, because they're bringing in with their roots some microbes. And each plant, there's dynamics that happen with different plant species, with different microbes, and it's very complex. And so the idea is, you're trying to build up that diversity over time and not have just single either plant species or microbe species. You really need that diversity to be effective. When you introduce a product with the plant, the idea is you're just helping that plant to have better quality, better yield. You're not trying to change the whole field. Changing the whole field takes a lot of management and you got to do a lot of different things, right?
Is this a fair analogy? I get sick, I take penicillin. Suddenly my body has all this penicillin in it from the tablet that I'm taking. But when I stop taking the penicillin, like that penicillin is not now reproducing itself in my body at those high levels. Maybe there's some that lingers there, I don't know. But like when I take the overload away in the form of a pill, maybe there's some residual, but it's not set up to live in me the way it would
Right.
in that tablet.
Well, and it's a good analogy in the sense of our bodies are healthiest, right, when we have a diversity of bacteria in our system, right? Our biotics. When we try to introduce probiotics, it can help improve your health for sure, but it's really that natural diversity and through, health and healthy habits that you can maintain it, right?
Mm-hmm.
When you try to have to introduce more, it can help locally, but it's really that diversity that maintains that health. And the same thing carries over, to soil and plants as it does in our bodies. So, probiotics certainly can help target and if you have a pathogen and, introducing an antibiotic certainly can help with that pathogen. But it really is maintaining that healthy system. And that's really where, like, the whole regenerative agriculture, soil health is headed is, we want to get that system back, right? That we've kind of taken away over time with some of our practices.
And I guess in a fair way, I mean, we don't want bad bacteria to be overloaded in the soil. If you think about it, sure, we would love to have as many of these growth enhancing bacteria in the soil as possible, but how much can the natural balance actually support? And so the system can go out of whack either way,
Yes.
and it can only support one base level.
Yes.
So if we circle back all the way back and we look at the research that was done at OSU when 3-bar came out, what were some of the specific bacteria and organisms in the soil that proved to be interesting and said, wow, okay, there's something here. This needs to be worked on more. And then those were identified, and hence 3-bar was born.
Yeah. So most of that early work, and mind you, the folks that are doing the discovery work, this is sometimes decades worth of their research, right, to find certain strains or groups of strains. And so that work had been going on for a good 10 years by the time 3-bar came along. Largely in that work, there's a grouping of beneficial bacteria from the genus Pseudomonas. And if you go into the research literature, many Pseudomonas species have been identified as being good growth promoters. And part of the reason is because they take a lot of different food sources, so they're not real picky. They're rapid growers, so we call them good root colonizers. So when the plant root system's coming out, that they grow really well, and they're not so picky about the plant species as well. So they're good generalists in that sense, and they're found everywhere in the soil in particular. And so that's the environment they know. So they're good, they're good candidates, but they don't form spores. And so that's where, just like Ohio State discovered, this range of Pseudomonas species, many other examples of that from both university and industry. Very few products have made it to market because they just don't do well when you try to scale up, manufacture, and then eventually get them distributed. They don't do well. So most companies don't put any effort into it because it's gonna be too hard, right? So that's where we decided, okay, let's figure out a way to do this. And so that was where the technology that we developed, which is where the microbes are actually grown at the point of use versus having to go through. So it basically allows you to get around the issues of being in the supply chain. So they're fresh. What the farmer gets is a fresh population, just like what the researcher was doing in their lab, a fresh population. That's kind of the way that we've approached this, which is very different from kind of the traditional way to manufacture these types of products.
Right. And that's your live microbe technology. And can you walk people through what that would look like if they had that on their farm? They can go to the website and check out what it actually looked like, but to visualize for them, describe what it is.
Yeah. So basically we talked about, microbes when they're in a dry form, they kind of go dormant and they stay fairly stable for a period of time. So that's what we start with. We start with the bacteria, but we only need a small amount. That is what we put in our cap system as part of this kind of live microbe technology. And then the other part of the system is liquid and nutrients. So we keep them in 2 separate chambers, and it's actually, when you're ready to use the product, you activate it, which means we've made it simple to use. So it's basically a push of a button, but then it allows the dry formulation, which has been stabilized, to combine with the liquid and nutrients. And so when that happens, basically that's when the fermentation starts, because the nutrients kind of come back out of that dormancy and they start to grow because they now have nutrients and liquid. And so what that looks like is it's basically a bag and box. So it's very easy for the grower to use, push of a button. But then you can pour, once the microbes have grown, which takes about 24 hours to go through a fermentation cycle, then they're ready to use. It's all contained. So there's no, we've produced it so that there's no contaminants. It's contaminant free. You're growing, you push the button, the microbes over 24 hours grow, and then it's ready to use. So you take that liquid and then you can apply it. You can, depending on your system and how you plan to apply it, you can deliver it as a liquid. So it's very easy to use. The microbes are 100% viable to start. They haven't been produced way upstream and gone through viability loss over time. So you're starting with a very fresh population. So for these gram-negative microbes, like Ohio State had discovered, this is a big deal, because otherwise trying to get them into to be viable by the time the farmer's using the product is a very big challenge. So this is kind of a way to do that with our system.
Because otherwise you're shipping them a a jug, probably with dry ice or something, right, to keep it cooler.
Well, one workaround, so the technology that we developed is a workaround for this, right, where you're growing them as close to the point of use as possible. Another workaround is refrigeration, right? So yes, you can produce them and then keep them refrigerated and ship them under refrigerated conditions. That's very expensive, not necessarily easy, but it does prolong their life to be longer than at room temperature, or worse if you get under elevated temperature conditions.
And the main product that 3 Bar has is Biostimulant. Can you talk about what that product is and what's in it, what it would be used for?
Yeah, so these plant growth promoting bacteria that we've been talking about, they can fall under what's called biostimulants or biofertilizers in some cases. In our case with BioYield, which is the first product that we commercialized from the strains we were working with Ohio State, we know that these bacteria, they produce, like I said, they produce a range of metabolites, so these chemistries. Some of them help kind of the plant through this range of abiotic stresses. In particular, we see, this cooler, wetter soil can really help boost growth of the plant through the early stages of growth in those conditions. It also produces organic acids, which can help with phosphorus solubilization. So that's one area. Any nutrient solubilization in the soil it can help with, but phosphorus in particular.
And is it primarily then Pseudomonas based, I guess?
Yeah, it's a Pseudomonas species. Yep.
Got it. And
Yep.
the liquid that you're using to breed it out, is that, something just like a sugar water? I mean, simplified down, but is that's carbon source?
Yeah, it's a carbon. Definitely, microbes need carbon and nitrogen, and then a range of salts. Most growth media that you use in the lab, you can different microbes grow best on different media. We can optimize that in our system, but a good nutrient-rich media that you would use in the lab, microbes.
When looking at this, there could be a variety of farmers. Somebody who grows corn, somebody who grows lettuce, somebody who grows strawberries, somebody who grows tomatoes. Could any of those growers look at this product and say, how could this help me? Or is it really targeted towards a specific crop?
And we definitely have started to collect more data on different crops. The bacteria, these type, what I call kind of, I hate to call them generalists, but the bacteria that are known, like Pseudomonas, that are known for associating with plants, they can provide a range of benefits that are kind of general. The abiotic stress helping with nutrient uptake, and that these Pseudomonas are very, like I said, they're not picky about their carbon necessarily. So they do associate with a wide range of different plant species. That's not always the case, but in some cases, like with some of these Pseudomonads, it is. And so you could use them on, and we have been collecting data, strawberries. We have some data, other specialty crops, tomatoes. So we continue to work it out with different plants. But we started with corn and soybeans because that was kind of where Ohio State started. So that was kind of where we first went commercially.
And right now, what's the optimum application point? Is it in furrow when you're planting? Is that where you see the best use of this product?
That's a great question. And that's something where we have started is applying it with the seed at planting, so that as that seed germinates and the early root system forms, the bacteria right there to grow with it, right? When you're adding the bacteria to an established root system, that's something that we're actually investigating right now to see how that looks compared to when you're applying it with the seed. And the thing to remember, with a precision application with the seed, you've got a lot of microbes right around that seed. So as the root system comes out, you can the bacteria can heavily colonize it. When you're doing like, drip irrigation, things like that, where you may have a point source, or even like a soil drench, you may not be getting the microbes close to the root system. They might be a couple inches away. With a soil drench, if you're spraying down or doing something like that, now you're diluting a lot of the bacteria across a wider area, so you may not have as many to start with that the roots will interact with. So things to think about around delivery to help these products work better. I mean, ideally, they're made to if you're putting them in the soil, they're made to interact with the plant in the root zone, right? So you want to have the bacteria at a high concentration in that root zone, regardless.
Right. For crops that are transplanted, if you look at lettuce and broccoli and stuff like we have out here in California, other
Yep.
crops like that, would it make sense to apply this in a nursery setting where you can be hyper focused? You can either apply it when the seed goes down, or just on each little cube of soil that you have right there.
Yeah, no, I mean, I think the earlier, especially for crops that are kind of vertical and they're going to go through a cycle, applying them probably multiple times, but early, either with the transplant at a young stage, during transplant. It just depends on, growers don't want extra steps added, so it has to fit into existing practices, right? So if there is, in plugs, you can certainly drench it in. It's right there. It's a small volume that can work really effectively. You just have to make sure that as the plant goes along, you're if you're going to repeat applications, that it needs to be near that root system and not, otherwise you might be wasting material.
Sure. With so many microbial products out there and so many people thinking of the upside and yet worrying about the downside and the cost benefit trade-off, what are questions you think people should really ask or information they should look for before they seek out a microbial?
Yeah, and it comes back to, what do you really need? I think for some of the growers in California, there's those needs are different than, row crop farmers in the Midwest, right? So look for products that can kind of meet those needs. For instance, if drought's an issue or trying to reduce water, there are microbes that are coming along, and I think some on the market, that can help the plant through that abiotic stress. If watering's intermittent, they can help maybe reduce the amount of water needed. There's bacteria that can help the plant through some of those conditions. That's a need, right?
Mm-hmm.
Nutrient, kind of the area where there is a lot of focus right now is in nutrients, right? Reducing particularly nitrogen and phosphorus, so that we don't have the runoff and contamination issues from fields. So there's a lot of research going on around nitrogen, being able to fix nitrogen from the atmosphere. So there's bacteria in nature that do that. But the ability to kind of amplify that and make it work. So there's a lot of new microbes coming out, companies spending a good bit to develop better microbes to do that. I think one of the things I would caution, there's products out there that have microbes in them. It mostly have, they've been produced and have metabolites in them, probably, that the microbes produced during their fermentation. Those products, whether the microbes are alive or not, they contain a lot of metabolites that can provide benefit. So these are bioactive compounds that the microbes produce, and you can see benefit. But for a living microbe product, you do want the microbe to provide kind of extended benefit over time if that microbe's added to your system, not just the chemistry that it produced. Make sure you understand, are the microbes, what level are they at? the regulatory framework for these products is coming in the US. It's a little bit behind some Europe and some other places around the world, but we don't monitor the quality of products yet. So there are microbe products that are supposed to have living microbes in them, but they don't, or they have, you know, contaminants in there or other species that are in there that shouldn't be. So the regulatory framework for that's coming, but just be mindful of, you know, try to understand what's in your jug, right? That it may not be completely living. One of the tests is if it stinks really bad, you probably have microbes that are on their way out because they're on their, you know, they're dying. So it shouldn't be a bad smelling product if it's fresh and viable. So just some things to be mindful of when you're using these products.
It seems like a buzzword for a lot of products, and it seems like a way that is for people to sell a lot of product. And, you know, I like what you said about, you know what you're actually getting. Is it living? Is it the byproducts of those microbes? Do you have any sense of how many bad players are out there? Not necessarily intentional, but given that there isn't a framework to say, here's what it has to be. And we can say they're in there and there could be one or there could be 1 billion, but one is still in there. Do you, working in this range, do you feel like there is a lot of latitude around what these microbial products are where people could find themselves in danger of buying something that's more or less just junk?
Yeah, and honestly, that's been the issue with this industry is the perception of snake oil, right? You know, people don't really know what's in the jug, and that's a problem. But what I can say is, I think where we are now, and there's a lot of science behind the products that are, you know, newly on the market, that have been on the market more recently, that, you know, as a regulatory framework comes along, that's going to be the standard, right? Um, I I know some of this. There's been a couple of studies that have looked at product quality. You know, the Department of Agriculture in Oregon, I think, has taken a big lead in monitoring products in their state. And some of the data that they have coming out of that work is less than, I think it's less than 40% of products had, you know, were meeting their label in terms of numbers of the viability numbers, as well as, you know, you said you were going to have these species in here and you don't, right?
Right.
So quality does come down to, you know, meeting your label limits of having so many viable cells in there, as well as making sure that you have only what you say you have in there, right? You don't have contaminants, which is, as you scale up manufacturing processes, that becomes more and more difficult, right, to maintain not having contaminants in there. And so right now the data doesn't look good. As an industry, it doesn't look good for us, right? But what I can say is there's many companies out there that are working to, you know, we're working on innovation and technologies to do way better than kind of what the data says right now.
So say I go to 3 Bar and I end up going with your tech. I get it on the farm. I apply it. Realistically, how should I focus my lens so I have appropriate expectations?
So I think two things right out of the gate is, you know, what you're applying is fresh, viable, because you just grew it. So you have that at least, you know what's in your jug, right? From that standpoint. And then being realistic about what it's meant to do. And so, you know, certainly our label, the early vigor and, you know, with the microbe we're using right now and helping with nutrient availability and uptake. So don't expect, I think, more than what the product's supposed to do.
Yeah.
And make sure it aligns with what you need, I think, are two ways to kind of level that expectation. and then try it. And I think that's been our biggest thing with, at least where we've kind of done some of our proof of concept work with corn farmers in the Midwest. We've had some farmers that have found a fit on some of their acres and continue to buy it year over year, and some that haven't really seen probably the level of benefit. And it comes back to, is it really meeting a need you have, right? So that's kind of where we start with our growers, is making sure that there's a right fit, have them try it, and usually try it for a couple years because of the variability you get year to year with climate and so forth.
And if somebody did have a question about fit usage, do you have people that they can call up and talk to and say, hey, here's what I'm trying to do. Here's what I have. One other thing is, along that line of farmer observation, is that a big key to really advancing this industry of, you need some farmers who are willing to kind of go out on a limb, take a chance, and do their own split testing so they can see, does this work? Does this not work? I'm going to compare it against, I'm applying it on plot A. Plot B is very similar in terms of soil structure and everything else. And I'm going to look at A versus B and just see what happens here. I feel like anybody doing any amendments of significance that offers some sort of benefit like this, the only way you're going to convince yourself at the end of the day is by looking at, okay, a control and then applied.
Yep.
Do I notice a difference? Do I see a difference? Do I taste a difference? Whatever that variable is.
Yeah, for sure. And that's where, I think the farmers that are doing that by, they're trying different things. They're kind of pushing, looking at new technologies. I think right now where we are with biologicals, that's mainly the farmers that are kind of using them and experimenting with them. And it's going to take, we're at the early stages of technology adoption, right? So it's going to take some of that experiment on the farm to see where it fits, how it fits. One of the struggles I have is, we're not going to have a market unless farmers are buying these products, right? And so, there's farmers trying them, some early adopters, but the research, what we haven't seen necessarily with, there are some university researchers, agronomists, working with these products, but not a lot. Extension has historically kind of been where some of these new technologies get vetted out. And, just kind of the nature of funding and, there's not a lot of research going on. There's more coming down the pipeline within universities, but not a lot. But I think farmers rely on that research too. So I think more of that in the U.S. is needed. More funding for the universities to do that, I think, will help strengthen the industry at large and give farmers maybe a little bit more confidence in some of these products. But otherwise, it really is the farmers trying things, but then also the industry carrying a lot of the load of industry, both companies and distributors doing a lot of this testing.
Right. Just looking in your crystal ball here, if you looked ahead 10 years, seeing how everything's advancing on the day-to-day, what do you think the biological microbial space and ag is going to look like?
Yeah, I think I'm just amazed at, technology is, moving fast right now because we have these tools to utilize, right? Biotechnology tools, things that have evolved very rapidly over the last 5, 10, 15 years. So the ability to discover and the speed of that is quickening. I have an example I've used in a presentation before, looking at kind of over the last 20 years. It took, the screening processes of naturally occurring microbes, isolating them, work that like Ohio State did and other universities have done, screening out, figuring out ones that work, timelines of kind of 15, 20 plus years to get a product to market. And what we're seeing now with, some of the industry players and applying some of these biotechnology tools and genetic engineering tools, that timeline is shrinking to less than 10 years to do that foundational work, get a candidate tested out and get it to market. So I think we're going to continue to see the acceleration of technology in this area, biotechnology tools being used. And then the big one that I think maybe the step changes that we're hoping for is when you start to bring the plant and the microbe together more intentionally to provide that bigger advantage. So you're basically doing the biotechnology work both with the plant and the microbe or microbes, multiple strains, to really create a system. I think that's where the next 10 years, we'll be talking about it more holistically as a system. And that research is underway in quite a few places.
Yeah, interesting stuff. And for somebody who likes what you're talking about here and likes what you guys have to offer, what's about the rough cost per acre of application at this point?
Yeah, it varies. It depends and it varies. One of the things we have always strived for is return on investment for the farmer, and so we've focused on keeping costs, trying to keep costs down. And so for at least what we're doing with our product, with a precision application, right? So it's going with the seed at planting. It's roughly between $4 to $5 an acre. So it's fairly low cost input for a row crop farmer. But products, certainly can get into, the 20 to $30 per acre range as well.
Got it. And if people want to learn more about everything that you guys are doing, where's the best place that they can go?
Yeah, I mean, our website is a great place, which is www.3barbiologics.com. If you're just interested in these microbial types of products and what they are, biologics in general, we do have a great industry association that has a good bit of educational information, and that's Biological Products Industry or Association, so BPIA. You can find that out on the internet, and just lots of great resources beyond that, so websites and whatnot that are, some universities that have some educational material out as well. So it's an area that's definitely, like I said, kind of at the beginning of we're utilizing technology. I do believe these products are going to get better and better as time goes on. And we're just really starting to understand, how to better use these products, how to better deliver them to make a difference. So if you are interested, certainly start to do some research and think about, the importance. Don't forget the importance of how you're delivering it, because you do want to make sure that what you're putting in the field is going to be effective.
Hi, everybody. Thanks for watching. Subscribe here to get the latest from the show. Also, be sure to check out some of the great clips and watch the full interviews right here on In Search of Soil.